Hello, I am Fernando Gordillo González. I am going to present you this poster that is about a novel integration of single-cell transcriptomic studies for the characterization of sex differences in Parkinson's disease. Sex differences in Parkinson's disease have been deeply described in the scientific bibliography, since there are differences in most important issues of it, such as symptom development, progression, or treatment response. However, molecular mechanisms that cause these differences remain unclear. With this background, our main goal is to achieve further information about these sex differences and its underlying mechanism. To do so, we implemented an in silico approach to integrate data from different single-cell RNA-seq studies accomplished in brain samples with Parkinson's disease. Here we can see the workflow of this approach, which starts with a systematic review, where adequate studies were selected from the principal repositories, such as GEO. This review followed the Prisma declaration, specific for this type of analysis. Then, selected studies are analyzed individually for the subsequent integration. This analysis consists, briefly, in a quality control and normalization of the datasets, in order to make them comparable. Next, a cell type annotation that classifies each cell based on its cell type. In this case, we annotated the dataset with main brain cell types, since we only had brain samples. The last step of this individual analysis is the differential expression analysis, performed for each cell type identified. In this case, we made this double comparison in order to identify sex-based differences. Afterwards, individual results were integrated by an intersection for each cell type and performed a functional analysis in order to give our results a biological interpretation. This workflow leads us to the following results. It is worth to mention that 2 studies were finally selected for the integration after the systematic review and individual analysis. It is also important to clarify that our automatic multi-approach cell annotation only managed to annotate correctly three cell types in both datasets: microglia, astrocytes, and neurons. So the differential expression analysis was performed only for these cell types. After the intersection of the differential expressed genes along all cell types and sexes, we obtain various common genes for every possibility, as we can see in this table. A brief summary of what we obtained after performing the functional analysis is that overexpressed genes in women's Parkinson were related with synaptic process, whereas in men's Parkinson were associated with neurodegenerative process and neuroinflammation. These results match with information already published, such as higher neuroinflammation in men or the possible neuroprotector effect of estrogens. The conclusions that we can extract from this above work is that single-cell RNA-seq has great potential to observe more specific changes in disease such as Parkinson's disease. Introducing sex information is essential for advancing toward personalized medicine. During the progression of Parkinson's disease, there is a greater neuroinflammatory process in men, while in women there is a potentiation of neurotransmission, and this potentiation in resilient cell may explain the delay in the Parkinson's disease onset in this sex. These results have been obtained from previously generated data, highlighting the power of this type of in silico approaches.